Good afternoon, Shruti Reddy. Okay, so I think I'm audible, right? Okay, fine, guys. Before starting the class, just I will 1 minute. Okay, for the video, so I'm going to close the video. Okay, just we will go with the audio. Fine. So here you can see, guys. I will launch the poll. Kindly tell me whether I'm audible and screen is visible. Okay. Yeah. I will launch the poll. Kindly tell me whether I'm audible and screen is visible. Okay, okay. Thank you so much. Screen is visible, right? I've offered the video. Screen is visible, right? Okay,
Yeah, yeah, it's visible.
fine. 1 minute. Okay, fine, guys. In the today's class, we are going to talk about the photosynthesis in the higher plants. You know that this is the important topic you have to study. You will get minimum 3 to 4 questions from this chapter. Okay, fine. So let us talk about the photosynthesis in higher plant, guys. We are not going to the basic things. You all know that photosynthesis is nothing but the plant utilize the light and they will synthesize the organic compound in the light, right? So it is called as the photosynthesis. Let us study about that in detail. Okay. In the photosynthesis, starting we will study about the experiment, the early experiment. Okay. So here, why you have to remember this early experiment means this type of the you will get the MCQ type of question where match the following will be there. In that, they will give this type of the question, that is early experiment. Okay. For that, you have to remember this early experiment. It is in the case of this chapter also, in the plant growth and development also. Okay, fine. So here you can see 1 minute. Yeah, here you can see Joseph Priestley. Priestley hypothesized you all know that Priestley performed the experiment. That is, he took the one bell jar. There, he kept the mice and here you can see he kept the mice and the candle there. But after some time, what happened? The mice and candle both are releasing the carbon dioxide. So due to this, the carbon dioxide release, the environment became polluted, right? The environment became polluted, there is no oxygen is present. Because of that, the candle has been extinguished and the mice also has been died, right? The later Priestley did is he kept the mint plant inside that bell jar. Then what happened, he kept the candle also, then the mice also. After some time, the mice also living and the candle is also is there firing. So what happened here, what he concluded here means, the whatever the gases it has been released by the mice and the candle, that has been recycled or it has been taken up by the plant and it is releasing the fresh air, right? So that is given by the Priestley, that restore air by the mint plant, that is nothing but the oxygen, right? And the Joseph Priestley is the scientist who discovered the oxygen also. Okay, remember the year 1770. Okay. Then coming to the John Ingenhouse, what he did, John Ingenhouse taken the aquatic plant. Okay, he took the aquatic plant. He did the same experiment in the aquatic plant. He kept that plant in the light condition and in the dark condition. What he observed is in the dark condition, in the light condition he kept. Okay, in the light condition he found that a bubble kind of thing. A bubbles is coming. The bubbles is evolving from the plant in the water in the aquatic plant. That bubbles is nothing but the oxygen, right? He observed that bubbles only in the light condition, only in the light, were present in the dark. Then he concluded that the green parts of the plant that could release the oxygen. Okay, only in presence of the light. Okay, remember, only in light. Okay. Then coming to the Julius own side, he found that in the plant there is a green part is there, leaves. In the green part leaves, there is a chloroplast is there. In the chloroplast, the photosynthesis is taken place where it is going to synthesize the glucose. Glucose is going to be stored as starch. Remember, the scientist name is Julius Onsack. Then T.W. Engelmann, you can see here. T.W. Engelmann did the experiment. Here in the experiment, he took the one of the green algae. What is that green algae, guys? Cladophora. Cladophora. Okay, here you can see he took the green algae. He took the green algae. Here you can see. I will explain you once again. Okay, he took the green algae. Kindly see this once again, guys. He took the green algae. Okay, that is cladophora. Okay, on this he kept a one. You can see one or two aerobic bacteria. Okay, this is aerobic bacteria. You know that this aerobic bacteria will grow only in presence of oxygen. Okay, aerobic bacteria. And what is this? Cladophora. Cladophora. Okay. And what he did is he took the one prism. Okay. He took the prism and he split the white light. White light is entering the prism and the white light is splitted. Okay. You know that the white light is splitted as the Vibgayar. Vibgyor. Right? It is splitted like the Vibgayar. Then what he observed is, okay, what he observed is the all the light is entering right here, violet light and violet, blue light, green light. These all the things is coming right. Yellow light, orange light, and red light. He observed that particularly the cladophora is doing the photosynthesis in blue wavelength and the red wavelength. Okay, blue wavelength and in the red wavelength. So due to this, the oxygen is going to be released only in this place where the blue wavelength is coming right there and the red wavelength is coming right there. Only in these two regions, the oxygen is evolving and the bacteria is accumulated only in these two region, where in the red region and in the blue region, not in the any other region, because in any other region there is a cladophora is not releasing the oxygen in any other region because it is not getting the particular light wavelength, right? So here in the blue wavelength and in only in the red wavelength it is getting the it is undergoing the photosynthesis. Then T.W. Engelmann concluded that the absorption spectra of chlorophyll A and chlorophyll B. Okay. The first absorption spectra is given by T.W. Engelmann. First absorption spectra. First action spectra is given by T.W. Engelmann. Just remember that. Okay. So here you can see the bacteria grown only in the blue and the red region. Then coming to the next experiment, Cornwall-Neal. Cornwall-Neal is taken the two bacteria. One is the purple sulfur bacteria, purple sulfur, purple sulfur bacteria and green sulfur bacteria. Green sulfur bacteria and the purple sulfur bacteria. By he seen that during the photosynthesis, this green sulfur bacteria and the purple sulfur bacteria is not releasing the oxygen. It is going to release the sulfur dioxide. It is going to release the sulfur dioxide because the hydrogen donor is different. Because hydrogen donor is H2S. Okay. Hydrogen donor is H2S in the purple sulfur bacteria and green sulfur bacteria. Whereas in the plant, which is the hydrogen donor, guys? Can you tell me which is the hydrogen donor in the plants? For the photosynthesis, which is the hydrogen donor in the plants? The hydrogen donor in the plants is H2O, right? So that's why the plant is going to release the oxygen. Whereas in the green sulfur bacteria and the purple sulfur bacteria, the hydrogen donor is H2S, right? So that's why it is going to release sulfur dioxide, not oxygen. Okay. So this is concluded by the Cornwallis-Neal. He told that the hydrogen from the suitable oxidizable compound reduces the carbon dioxide to carbohydrate. Okay. Hydrogen donor is different in different organisms. So example, in purple sulfur bacteria, the hydrogen donor is H2S. In the plants, it is H2O. This is about the early experiment. Have you understood this? This is a brief. Do you understood this, guys? In a short, I have told the experiment. Can you give me thumbs up, or did you understood? Give me a message. Did you understood this all the experiments? Okay, fine. Okay, then coming to the photosynthetic pigments, guys. So, which are the photosynthetic pigments are there? There are mainly chlorophyll A, chlorophyll B, and xanthophyll. Xanthophyll. 1 minute. 1 minute. Yeah, yeah. Here I will write. Okay. Chlorophyll A, chlorophyll B, and xanthophyll. Xanthophyll. Then carotenoids. Carotenoids. Okay, carotenoids. Here you have to remember, guys, chlorophyll A, chlorophyll A absorbs in both blue and red wavelength. Okay, it absorbs in both blue and red wavelength, and it is having the color is a bluish green. Okay, it is having the a bluish green, or we can call it as greenish blue. Okay, it is having the bluish green color. Okay, then coming to the chlorophyll B, chlorophyll B, it is having the yellow green color, yellow green or olive green. We will call it as olive green. Okay. Then coming to the xanthophyll, xanthophyll is yellow color. Okay. Then coming to the carotenoid, carotenoids are yellow to orange, yellow to orange. So that's why the carrot is having the carotenoid. It is orange in color. Okay. So remember these things. Carotenoids is yellow to orange color, xanthophyll is yellow color, and chlorophyll B is yellow to green, that is olive green color, and chlorophyll A is bluish green. Okay. The main pigment is chlorophyll A. Okay, chlorophyll A is the main pigment, whereas chlorophyll B, carotenoids, and xanthophyll. These three is acting as the accessory pigments. Accessory pigments. Kindly remember this. Okay. Chlorophyll A, sorry, chlorophyll B, chlorophyll, this is carotenoids and xanthophyll. These all are acting as the accessory pigments. I think you all understood this about the pigments. Let us talk about the site of photosynthesis. You all know that where is the photosynthesis going to be taken place? It is going to be taken place in the chloroplast. Right. You have studied chloroplast in the cell chapter. Chloroplast is double membrane, is having the two membrane, right? Outer and inner membrane, right? Then inside this, the fluid-like structure is there. That is going to be called as stroma, right? In the stroma, the dark reaction is going to be taken place. Dark reaction. Kindly remember this, guys. Dark reaction is going to be taken place in the stroma, whereas the light reaction is going to be taken place in thylakoid membrane. Remember this, thylakoid membrane. Okay, only this much is enough. Okay, I think you know the structure. The chloroplast is having own its double circular, double circular DNA and 70S ribosome. You all know this, right? No need to explain once again. Okay. There is a structure where the dark reaction and light reaction is going to be taken place. Now let us study about what is action spectra and what is absorption spectra. So here you can see, guys, this is the wavelength. Okay. In the x-axis, they are given the wavelength. Okay. Wavelength is here in the x-axis. And in the y-axis, they are given the absorption. Absorption. Okay. Absorption. Remember, on the y-axis absorption, on the x-axis wavelength. Here what they are doing is, here the pigments are there, right? Chlorophyll A and chlorophyll B. Here you can see a green color, a bluish green color is there, right? Bluish green color. It is nothing but the chlorophyll A. And a green color, a pale green color is there, right? That is nothing but the chlorophyll B. You can observe there are the four pigments are there here. They are given three pigments mainly: chlorophyll A, chlorophyll B, and carotenoids. Main pigments are chlorophyll A and chlorophyll B, right? Both are absorbing the light at around 400. That is in the, 1 minute. That is in the blue wavelength. Here the 400 will start, okay? From here 600 will start. Here you can see in the downwards, okay? The mainly the chlorophyll A and B is absorbing in the blue wavelength. Here you can see here also you are getting, and again in the red wavelength also you are getting, right? Means chlorophyll A is having the absorption at both blue and red wavelength, okay? Both blue and red wavelength, okay? Then coming to the chlorophyll B, guys, here you can see that is a green color. Chlorophyll B also is absorbing at the blue wavelength and in the red wavelength. In the red wavelength, it is less. You can compare to the, in the exam, they may give you diagram. How can you differentiate is chlorophyll B is having the less curve. Here you can see a less hump compared to the chlorophyll A. Okay. So the in the red wavelength, the photosynthesis is also going to take place in both chlorophyll A and chlorophyll B. Absorption spectra is nothing but a curve it is giving where when the pigments absorb the light. That is nothing but the absorption. Sorry, that is nothing but the absorption spectra. Okay. Then coming, I will give you the example, guys. It is nothing but if I'm teaching to you, right? Now I'm teaching to you how much I'm teaching to you, how much you are understanding. Okay, now you all are pigments. Okay, now I'm teaching lesson to you. How much you are going to understand? It is nothing but the absorption. How much things or how much lecture is going to your mind? That is nothing but absorption. Okay, let us talk about the action spectra. Action spectra is nothing but how much I did the lecture to you, how much I explained the topic to you, how much you understood. It is nothing but absorption spectra, right? How much you understood and how much you written in the exam. How you performed your exam, no? How the result will come, right? How the result is coming. That is nothing but the action spectra. Action, what action you are taken. Action spectra is nothing but the result. Okay. See here, the mainly the absorption spectra is happening blue and red wavelength, right? Only in the blue and red wavelength you can see this is nothing but the action spectra. Action spectra is nothing but the photosynthesis. Photosynthesis taken in particular wavelength. Okay, the photosynthesis taken in particular wavelength is called as action spectra. Okay. Together you can see the action spectra and absorption spectra here. This here you can see that here they are given the action spectra here and here they are given the absorption spectra. Both are coinciding where both are coinciding in the blue and red wavelength. In both blue and red wavelength, the absorption spectra and action spectra is showing the rise means the specific pigment is absorbing that wavelength and specific photosynthesis happening in that wavelength means that pigment is important for the photosynthesis and that wavelength is important for the photosynthesis. Only in that wavelength the photosynthesis is going to take place. Okay, that is mainly in blue and red wavelength. Do you understood this? Kindly go through this in the NCRT book also because we are doing a crash course. I can't explain you all the things, guys. That is actually the problem. Okay, fine. So let us go to the light reaction, guys. You all know that what is the light reaction, right? The light reaction doesn't mean that it has to be taken place only in the light, right? Because it has to taken place in the light only because you know that only in the light the water is going to be split and also the light is going to be absorbed, right? For the pigment. Here you can see, guys. The light reaction mainly include light absorption, water splitting, and oxygen release and formation of the ATP and NADPH. There are the two reaction is there, right? Light reaction where it is light dependent. Light is important. Whereas the dark reaction or photosynthetic phase, photosynthesizing phase here, mainly the organic compound is going to be synthesis here. It is light independent. Light independent, right? Okay. Doesn't mean that dark reaction will take place in the light. The dark reaction will take place in the light also, but it require the product of light reaction, not light. Okay, fine. So let us see here, guys. Mainly the steps are taken place in the light reactions are, first is light absorption. Here you can see, here I have given here. This diagram is important. Okay. These all are the accessory pigments. Accessory pigments, which all are the accessory pigments, guys? Chlorophyll B, carotenoids, and xanthophyll, right? These all the pigments will absorb all the pigments will absorb the light and it will send to the primary acceptor or to the main reaction center, that is a chlorophyll A. Okay, it will send to the main reaction center, that is a chlorophyll A. Then the electron is going to be passed. Okay, so that is mainly the light absorption. Okay, after the light absorption, the water is going to be split. Water is going to be split here. You can see that two water molecule is splitted here. It is going to convert 4 proton, oxygen and 4 electron. Okay. Now this 4 electron is going to be move here. You can see this 4 electron is going to be move in a reaction. Okay. Then coming to the oxygen release. The oxygen release is going to be taken place in photosystem 2. Okay, in the photosystem 2, the oxygen is going to be released. This oxygen is going to be released. And the formation of the ATP and NADPH is going to be taken place. And here you can see that the water splitting complex that is present in photosystem 2, that is located on the inner side of thylakoid membrane. It is very important. In the statement wise question, they will ask you in the light reaction, it is important. The inner side of the thylakoid membrane. This is the thylakoid membrane, right? The inner side of the thylakoid membrane, the photolysis is going to be taken place. Okay, fine. So here you can see in the exam, they ask about what are the products. What are the products of light reaction means oxygen, okay, oxygen, ATP and NADPH are the products of light reaction. Okay, kindly go through this. Okay, so let us talk about the cyclic and non-cyclic photophosphorylation. What do you mean by cyclic photophosphorylation, guys? Can anyone tell me what do you mean by cyclic photophosphorylation? Okay, so first let us learn about the non-cyclic photophosphorylation. Then we'll go to the cyclic photophosphorylation. Okay, so here non-cyclic means the electron which is cycling here, the electron which is cycling here does not return to the same molecule again, does not return to the same molecule. So that's why it is called as non-cyclic. Electron will not return again. Okay, so that's why it is called as non-cyclic. Okay, so here you can see that in the non-cyclic, it will look like this Z scheme. Z scheme. Here you can see it is look like this Z, right? This is a non-cyclic. Here what is happening is the water is splitted, the light is absorbed here, right? Light absorption has been taken place. The water splitted, the electron is there, right? Now, the light absorbed by the light harvesting complex. Now, this light harvesting complex transport the electrons. It will transport the electrons to the primary acceptor. Primary acceptor in the Z scheme is the pheophytin. Pheophytin. Kindly remember, pheophytin is the primary acceptor. It will send to the primary acceptor, then to the, you know that there is a cytochrome BCF complex is there, right? It will transport to the electron transport system. Then from the electron transport system, it will go to the light harvesting complex, that is photosystem 1. There is a two photosystems are there, right? From the photosystem 2, it is transported. That photosystem 2 is the 680 nanometer and photosystem 1 is the 700. Okay, now the light is absorbed. The electrons are splitted here. The electrons are there here. Now the electrons will be transported by the electron harvesting complex to the primary acceptor. Then it will go to the electron transport system. Then it is go to the photosystem 1. From the photosystem 1, it is going to the again electron acceptor. Then the final acceptor of the electron here, because the electron is going to be not returned, right? So final electron acceptor here is NADP plus. The NADP plus is going to be accept the electron. then it is going to be converted into the NADPH. NADPH, it is going to be reduced. Okay, the electrons is going to be received by the NADPH. In the exam, they may ask you which is the final electron acceptor. Final electron acceptor means that is NADP. Okay, remember, not H. H is the reduced form. NADP plus is the oxidized form. Okay, it is going to be accept. This is a non-cyclic. Okay. Here in the non-cyclic, we have seen two photosystem are involved and photolysis of the water is going to be taken place here. You can see this, and oxygen is going to be evolved, right? When the photolysis is happening, obviously the oxygen is going to be evolved, right? Then coming to the NADP, H and H plus is synthesized, right? Then electron does not return to the reaction center. This is about the non-cyclic photophosphorylation. Then coming to the, do you understood guys what is the non-cyclic photophosphorylation? Do you have any question over here? Can you tell me whether you understood or not? If you have any question over here means I will explain you once again. Non-cyclic photophosphorylation. Guys, can you tell me whether you understood this topic? Deepu told yes. What about others? Athine, Dini, Divya, Helen. Very good, Shruti Reddy. If you didn't understood means tell me guys. Once again, I will explain you. Because these all are important topics. In the exam, they may ask you the questions like this. In the non-cyclic photophosphorylation, both the photosystems are involved. Yes, both photosystems are involved. Is that ATP and NADPH is synthesizing there? Yes, both NADP and ATP is synthesizing there in the non-cyclic photophosphorylation. Is water is splitting? Yes, water is splitting. Is oxygen is releasing? Yes, oxygen is releasing. It is exactly like the photosynthesis mainly taken place in the non-cyclic photophosphorylation. Okay, whenever the light intensity is less, okay, when the light intensity is less, on that time, it is going to be undergo cyclic photophosphorylation. Okay, it is going to be undergo cyclic photophosphorylation. And what happened in the cyclic photophosphorylation, guys? Mainly, in the cyclic photophosphorylation, it will take only sometimes. Okay, okay. In the cyclic photophosphorylation, only one photosystem is involved. That is photosystem 1, that is 700 nanometer, that absorb in the 700 nanometer, right? Then there is no photolysis of water is taking place because you know that the photolysis of water is taking place only in photosystem 2, right? Here it is only one is involved. That's why the photolysis is not taking place. If the photolysis is not taking place means the oxygen is going to be evolved. No, the oxygen is not going to be evolved here. Yes. Then NADPH, 1 minute. Yeah. Then NADPH is synthesized here. The name of this is cyclic photophosphorylation. The same electron is recycling. Here you can see, guys. I will explain you here. See here. The light is absorbing here. Here also some always some electrons will be there. Okay. Always some electrons will be there here. Now the light is absorbed by the photosystem now. Right. Now it is absorbed the light means what it will do? It will just pass the electrons. Right. It will just pass the electrons to the electron acceptor. Then the electron acceptor, it will send the electrons to the cytochrome BC complex, right? You can see a cytochrome BCF complex. Then again, that electrons will be transported to the chlorophyll, that is the photosystem 1, right? Where the electron is cycling between the photosystem, electron acceptor, then electron transport system. Again that electron is cycling in the same manner. But what is happening here is the proton gradient is going to be formed here. Mainly the ATP is going to be synthesized. ATP is going to be synthesized. There is no synthesis of NADPH. Okay. There is no synthesis of NADPH here. Only ATP. Only ATP is going to be synthesized in the cyclic photophosphorylation. No NADPH, no splitting of water, and there is no involvement of the photosystem too. and there is no release of oxygen. Only ATP synthesis. That's it. That is about the cyclic photophosphorylation. Okay. What you have to do is you have to remember the diagram. They may give you diagram and they will remove the some of the labeling there and they will tell you to identify that by ABC. Okay. You have to remember this, guys. Fine. I think you understood about the non-cyclic and the cyclic photophosphorylation. Now we'll go to the chemiosmotic hypothesis. Chemiosmotic hypothesis of ATP formation. Okay. You know that, guys, the chemiosmotic hypothesis is given by Peter Mitchell. Peter Mitchell in the 1961. You have to remember the scientist name because in the exam they may ask you the scientist name. Okay. Here you have to see here, guys. This is how the electrons is going to be move. Okay. What I will tell you is just see this diagram now and just see this 3 point what I am explaining you. Okay. Then this concept will be easy for you. See here, guys. For the synthesis of ATP, now we are going to talk about there is a uh, there is a thing that is giving the how the ATP is going to be synthesized by the Peter Mitchell. He told how the ATP is synthesized by this theory, synthesis of ATP. The synthesis of ATP require proton gradient. See here, guys. For synthesis of ATP, we require proton gradient mainly. Okay. And here you have to see where is this proton gradient is going to be formed. Between stroma, okay, between stroma and in the lumen. Okay. You know that lumen is nothing but the center. And after that, every part we are going to call as the stroma only. Stroma, right. This circle is there, no? Inside white color part, that is nothing but lumen. Outside part, all outside part we calling as the stroma. Okay. And here, how is that proton gradient is going to be formed? You all know that. What is a gradient, right? From the lesser to the higher or from the higher to the lesser. That is nothing but the gradient. Here, what will happen? In the stroma is having less, okay? It is having less proton. It is having less proton compared to the lumen. In the lumen, there are many protons are there, more protons are there. Now you tell me, guys, in the lumen it is having the acidic pH or in the stroma it is acidic. Can anyone think, guys, whether the stroma is having the more acidic or the lumen is having the more acidic? Where the proton is there more, there the pH is more acidic, more acidic. Where more proton is there, there is more acidic pH. Then coming to the stroma is having the less acidic pH, less acidic, right? Now we have a question that I told you this is a gradient we required for the ATP synthesis. Lumen is having more hydrogen atom and it is having less pH, and in the stroma it is having less proton and having the more pH, more pH. I mean more acidic. Sorry, it is having the less acidic, right? Now the question arises here is how the lumen is filled with more proton. How the lumen is filled with more proton means we have a three things here. Okay, three steps mainly. See here, guys. These three steps are there. These three steps will tell you how this proton is accumulated inside the lumen and how this proton decreases the pH in the lumen. Okay. So mainly three things. These three things are very, very, very important. You have to remember. I will explain you. See here, guys. You all know that the photolysis of water. The photolysis of water is going to be released H and O. Right. Proton and oxygen is going to be released by the photolysis of water. Right. So now what will happen? This proton is there, right? Now this proton you can see on the thylakoid membrane, photolysis of water is going to be taken place, right? You know that oxygen is going to be released outside. Now, proton will be there, right? The proton will be sent inside the lumen, okay? By the photolysis of the water, the proton is releasing that will come into the lumen. That is the first step. Then coming to the second step, the electrons are also released, right, during the photolysis of the water. Now, this electrons is going to be transported, right? The electrons, 1 minute, 1 minute. Yeah. This electrons is transported from the photosystem 1, right, guys? Sorry, from the photosystem 2, right, guys? Then it will go to the primary acceptor, right? This primary acceptor is electron acceptor, but this primary acceptor will send to the electron. Here you can see it will send to the electron, but that is not electron acceptor, is the proton acceptor, is the proton acceptor. Okay, where the primary acceptor will send the electron to the proton acceptor, not to the electron acceptor. So what he will do is, he will take the proton from the stroma and he will send that to the lumen. Okay, we'll send that to the lumen. For example, you can see here, there is a membrane. Okay, so here photosystem 2 is there, and here primary acceptor is there. The primary acceptor, after the primary acceptor, here a proton acceptor is there. Proton acceptor. What he will do? The electrons is transported from the photosystem to the primary acceptor, right? Now, this primary acceptor will send the electron to the proton acceptor, not to the electron acceptor. So that's why what this proton acceptor will do is, okay, give me that electron, I will keep it, okay, but I will transport the proton from the stroma to the inside of the lumen. Okay. While passing the electrons together, what he will do is he has to do work also, right? So that's why what he will do, he will transport the protons from the stroma to the lumen. Due to that, the proton is going to be increases inside the lumen. This is the second. Okay. Now coming to the third one, the NADP reductase. You can see that the NADPH is there here. NADP reductase will be there. NADP reductase. It is the enzyme. It is required for the converting this NADP to the NADPH. What he will do is, he will convert this. He will take the protons where the NADP H plus is forming, right? This NADP reductase is faced to the stromal side. Stroma is facing towards the stroma. What he will do? He will take the proton from the stroma and he will send that protons to the lumen. Okay. Now, in the lumen, due to this, all the three things, by the photolysis of water and by the primary acceptor is sent to the proton acceptor, and due to the NADP reductase, the proton is goes on accumulated in the lumen. Okay. So now let us see what will happen. Okay. So now the proton gradient is formed. Right, guys. Now proton gradient formed. Proton gradient is formed. We require this proton gradient for ATP synthesis. Right. Now we will concentrate on how the ATP is going to be synthesized. Now we have all the proton in the lumen. Right. Now what will happen? The main enzyme is there. That enzyme is ATP synthase. ATP synthase enzyme that is located on the membrane, thylakoid membrane. It is having two subunit, guys. It is having two subunit. One is it is having two subunit. One is F naught and another one is F1. You know this F naught we call as a proton pump. Proton pump, and this proton pump, it is helping in the transport of this proton. And here this F1 is there. It is mainly acting as the ATP synthesis. It is mainly helping in the ATP synthesis. Okay. So now what will happen is, this molecule, this F naught, you can see the F naught, the proton will be passed from this F naught to the ATP synthase. It will send two proton for every ATP synthesis. Now, these two protons is transporting from the F naught. Now, the ADP plus PI is combining and it is going to be converted into the ATP. Okay, now this ATP is releasing. For every one ATP synthesis, we require two proton molecules. Remember this. Okay, guys. I think you understood about the chemiosmotic hypothesis. Once again, you have to go through this, guys. Okay. Mainly the important thing is proton gradient that is formed inside the lumen. Now for the ATP synthesis, the proton is moving from the lumen to the stroma. Remember, from the lumen to the stroma it is moving. Okay, fine. This is about the chemiosmotic hypothesis. Let us talk about the dark reaction, guys. Dark reaction is also called as the Calvin cycle because it is given by the Calvin Benson. Okay, Calvin Benson, he discovered this dark reaction by using the radioactive 14 carbon. Okay, and you all know that this dark reaction is going to be happen in the stroma. Okay, what you have to remember here is the main enzyme is Rubisco enzyme. This pathway, the dark reaction having mainly three processes. One is carboxylation, a reduction, and regeneration of ATP. Okay, this dark reaction is also called as C3 cycle. Okay, this dark reaction is common in both C3 and the C4 pathway. Why it is called as C3 mainly means the first product. The first product here forming is that is called as glyceraldehyde. Here you can see 3-phosphoglycerate. 3-phosphoglycerate. Very, very important. 3-phosphoglycerate is the first molecule, the first product. So that's why it is called as a C3. And the first acceptor. The acceptor is ribulose bis 5-phosphate. Okay, ribulose 1,5 bis phosphate. Okay, this is the acceptor. It is a 5-carbon molecule. Okay, this is acting as the acceptor. This is a 5-carbon molecule. Let us talk about the first reaction, guys. Carboxylation reaction. In the carboxylation reaction, the first acceptor molecule is ribulose 1,5 bisphosphate. This is going to be accept the carbon dioxide, and the enzyme is going to be help in the carbon dioxide acceptance is a Rubisco enzyme. Now this is going to be form a first stable product, that is the 3-phosphoglycerate. This is the important. Okay. Now this second process is going to be happen. The first process, carboxylation, does not require any ATP. Okay. Then coming to the second process, that is a reduction process that involving in the conversion of 3-phosphoglycerate to the glyceraldehyde 3-phosphate. That is a 2 molecule of glyceraldehyde 3-phosphate. Where these reaction, the reduction process, for each carbon dioxide fixation, it will require around 2 ATP molecule and 2 NADPH. Okay, for one carbon dioxide, that is in the reduction process. Remember. Okay, for only reduction process, 2 ATP and 2 NADPH is required. So totally for the C6H12O6, for the carbohydrate synthesis, for the glucose synthesis, we require 6 carbon dioxide. 6 carbon dioxide mean for the reduction we require totally 12 ATP molecule and 12 NADPH molecule. Okay. The products of the light reactions are utilizing in the dark reaction. Okay. So now you can see that the reduction process is completed. Now the third process is the regeneration of RUBP. This requires for the 1 carbon dioxide, it requires 1 ATP. 1 ATP molecule for the regeneration. So that's why for the 6 carbon dioxide, it requires 6 ATP molecule for the regeneration. So totally for the dark reaction, you can see here, 6 carbon dioxide is required because C6H12O6 is there, right? For the one glucose synthesis, it requires 6 carbon dioxide and 18 ATP and 12 NADPH. You know that 12 ATP in reduction and the 6 ATP in the regeneration. Okay. If they ask about for fixation of the one carbon dioxide, if they ask about the fixation of one carbon dioxide, how many ATP and how many NADPH is required means 3 ATP and 2 NADPH. Okay, if they ask about only in the reduction process means 2 ATP and 2 NADPH. Overall, they ask me 3 ATP. Okay, this is about the dark reaction, guys. Then we'll go to the Hatch and Slack pathway, that is a C4 cycle. You can see here, C4 cycle is mainly having C4 plant. Here we call as a C4 cycle because the first stable product, the first stable product is carbon 4. It is having the C4, that is nothing but oxaloacetate. Oxaloacetate. Okay, that's why it is called as the stable compound. Okay. Then coming to the here, the Kranz anatomy. All C4 plants are having the Kranz anatomy. What do you mean by Kranz anatomy? It means very simple, guys. Simple. See here, don't get confusion. Vascular bundle is there. The vascular bundle covered by a bundle sheet cell. Okay. So here you can see this is a vascular bundle. Okay. This is nothing but the vascular bundle. This vascular bundle is covered by, it is covered by bundle sheet. Bundle sheet cell. Okay. This bundle sheet cell is going to covered by mesophile. Mesophile. Okay. So this is called as Kranz anatomy. Okay. Just remember this is called as Kranz anatomy. Okay. And here one thing is there, guys. In the C4 plant, chloroplast dimorphism is there. Okay. What do you mean by chloroplast dimorphism means? Chloroplasts are different in structure. In bundle sheet cell, they are having the large chloroplast where the chloroplast does not have the grana. So that's why here dark light reaction is not going to be taken place in the bundle sheet. Only in the bundle sheet, there is a presence of Rubisco enzyme. Rubisco enzyme is present only in bundle sheet cell. The dark reaction is going to be taken place only in the bundle sheet cell. Remember, okay. Then coming to the mesophile. In the mesophile cell, the light reaction is going to be taken place because it is having the grana. Okay. It is having the grana. It contains the grana. Light reaction will take place. But there is no Rubisco. There is no Rubisco present here in the mesophile cell. That's why there is no dark reaction will take place in the mesophile cells. Okay, remember this. And here we will see about the C4 cycle. The atmospheric carbon dioxide is taken up by the 3 carbon compound, that is nothing but phosphoenol pyruvate, 3 carbon compound. This carbon dioxide is dissolved in the water, then it will converted into the bicarbonate. It is going to be accepted by the phosphoenol pyruvate. It is fixed. Okay, this is called as carboxylation. The carboxylation is going to be taken place here by the phosphoenol carboxylase enzyme. Then here it is going to be fixed into C4 acid, that is nothing but oxaloacetate. Okay, this oxaloacetate cannot transport to the bundle sheet cell. Now, this oxaloacetate is converted into the malate. Malate. Okay. This malate is transported here to the bundle sheet cell. Now, this malate is the C4 carbon, right? This is going to be release the carbon dioxide. Now, this carbon dioxide is taken by Rubisco enzyme in the bundle sheet cell. This is nothing but bundle sheet cell. The Rubisco enzyme will take the carbon dioxide, and here the dark reaction is going to be taken place. Okay. So now after the loss of one carbon dioxide, it is going to be converted into the C3 acid. Then again, it is going to be transported to the mesophile cell. Okay. So this is about C4 plant or the C4 cycle. The mainly C4 plants are the sugarcane, maize, and these all the things will come under the C4 plant. In the C4 plant, there is no photorespiration taking place. There is no photorespiration is taking place because the mesophile does not contain the Rubisco enzyme. The carbon dioxide is concentrated in the bundle sheet cell. That's the reason. Okay, fine. Let us go to the CAM pathway, guys. Nothing in the CAM pathway. What you have to remember here is the stomata. Stomata is going to be, CAM is Crassullian acid metabolism. Stomata is going to be open in the night. Okay, it is going to be open in the night in the, okay, whereas in the light condition it is closed. Okay, only have to remember this. Okay, then coming to the photorespiration, you all know that the Rubisco enzyme is having more affinity for the carbon dioxide, right? More affinity. But sometime what will happen, this Rubisco is have the affinity, the Rubisco will combine with the oxygen. On which condition? When increased light intensity. When light intensity is increased, the Rubisco combined with the oxygen. Now what will happen? This Rubisco combined with the oxygen means It is going to produce the two molecules mainly. What is that? One molecule of phospho. Here you can see that it is going to be produced the one phosphoglycerate, one molecule of phosphoglycerate, glycerate, and one molecule of phosphoglycolate, glycolate. Okay, this glycolate is two carbon molecule and phosphoglycerate is three carbon molecule. Okay, so this is a wasteful process we will call it as, and mainly three organelles are involved here: chloroplast, peroxisome, and mitochondria. Mainly in the question will arise from where is the glycine is synthesized in the photorespiration. Glycine is going to be synthesized in the peroxisome. Remember, three organelles are involved: chloroplast, peroxisome, and mitochondria. In the peroxisome, glycine is going to be synthesized, and in the mitochondria, serine is going to be synthesized. Please make a note of this, guys. Okay, fine. Let us solve some of the questions of the previous year, guys. Let us solve some questions here. Here is the question for you. Which of the following are required for the light reaction of the photosynthesis? Okay, 1 minute. I am launching the poll. 1 minute. Sorry, 1 minute. Yeah, I have launched the poll. You can give me answer in the message also. Very good, Shruti. Yes, Raju. Yes, Aicharan. Yeah, which is the correct answer, guys? As we are having the less time, we can't go in detail, guys. But whatever the things I've explained here, those all are very important for the exams. Okay. So that's why whenever you're coming for the revision chapter, for one time you have to read and you have to come. Then only you will understand clearly. Okay. Then that chapter will become easy for you. Okay. Without reading you are coming means it will feel very difficult. That's why whenever you are coming to the next class, you have to read the chapter, and if you come in while I'm we are doing the revision, it will be easy for you. Okay, I'm ending the poll, guys. 33% given A, and D is given by 67%. Okay, see here, guys. 1 minute. Yeah, this is asked in the 2024 re-examination. The correct answer is B, guys. B. See here, guys. We are talking about light reaction of photosynthesis. They're not asking you about the photosynthesis or dark reaction. See here. When we required for the light reaction, water we required. For the splitting of the water, we require the hydrogen. Yes or no? Right. Then coming to the chlorophyll, for the absorption of the light, we require the chlorophyll, right? Yes. Then coming to the light, we require the light for the photosynthesis, yes or no? Only for the light reaction, we require water, chlorophyll, and light. Why we don't require the oxygen, guys? Oxygen is a product of photosynthesis. Why we require this? We don't require this. Then coming to the carbon dioxide, here they are asking about the light reaction, but here the carbon dioxide is required for a dark reaction. That's why we don't require carbon dioxide. That's why the correct answer is D. But most of them is given A and D. Where you went wrong? You didn't see the question properly. Just you observed photosynthesis. But here they ask about light reaction. They're not asking about the product also. They are asking about what we require. Yes or no, guys? Did you understood this question? Kindly give me thumbs up if you understood means. Okay. Let us go to the next question, guys. Which one of the following product diffused out of the chloroplast during the photosynthesis? ADP, NADPH, oxygen, ATP. Just see the question properly. Which is diffusing out? What we don't require, which is releasing out? See the questions properly. The questions were very easy, but the understanding is important. Can you think once again the correct answer? Which is diffusing out? Which are the product of light reaction we studied, guys? Oxygen, ATP and NADPH. Yes or no? Very good, very good. Most of them are given the correct answer. The correct answer is oxygen. Oxygen is going to be released out. Right. Then here is the question, guys. Here you have to see that it is a diagram based question. You have to see this is a dark reaction. Dark reaction. Okay. So here you have to find this a process. Here they are given the process A, B, and C process. Simultaneously you have to see. You all know that there is a three processes are involved: carboxylation, reduction, and the regeneration. But what is the order? You have to see the order. Sometimes they will give A here, B here, and C here. Okay. On that time, you will just simply give the carboxylation, reduction, and regeneration. But that is not correct, right? On that time, what you have to give? Carboxylation, regeneration, reduction, because B is labeled here. Yes or no? You have to see properly. But for the here, the correct answer is C. A is the carboxylation, B is reduction, and C is the regeneration. Okay, C is correct. How many molecules of ATP and NADPH are required for every carbon dioxide molecule fixed in Calvin cycle? See here, total. They are asking about total. Total ATP and NADPH fixed in the Calvin cycle. Total they are asking. Very good, Dipu. Very good, Shreya. Yeah, I'm launching the poll. You can answer me in the poll, guys. Just to see, they're asking about overall reaction. Overall. Overall, they're asking. Okay, I'm ending the poll here, sharing the result. 25% given A, 25% C, 25, sorry, 50% D. Very good, guys. All are given D option. That is the correct. You can see here, guys. This is the dark reaction, right? Carboxylation. We don't require any ATP here. ATP and NADPH. We don't require here. Whereas the reduction process for one carbon dioxide molecule fixation, we require three. For the reduction, we require 2 ATP and 2 NADPH we require. Then regeneration, we require 1 ATP only. 1 ATP only. Then overall, we are talking about the Calvin cycle mean we require 1 and here 2 ATP, 3 ATP require and 2 NADPH we require. So the correct answer is D. Then here I am giving the match the following. You have to match. So we see here very simple question. This is also given in 2024 exam. Okay. So here you have to match the color. So many of you will get confused here. Can you tell me the answer in the chat box, guys? You can tell me your answer in the chat box. I launched the poll also. You can give me your answer in poll also. Quick. You can answer me in the poll. You can go through this PPT also, guys. There is so many questions are there. You can go to this. Okay. Fine. I'm ending the poll. I'm sharing the result. You can see here A is given by 67%, B is given by 33%. Let us match here, guys. Chlorophyll A. What about chlorophyll A, guys? Chlorophyll A is blue-green, right? Then A is 3. Can you match A with the 3? These two option is having A is the 3. Then coming to the chlorophyll B. Chlorophyll B is yellow-green, right? Then 2. Then where the B is having 2? Only in the A option, B is having the 2. The correct answer is A here. Then xanthophyll is yellow, carotenoid is yellow to orange. Right? Okay, guys. So this is about the today's class, guys. Kindly read once again the photosynthesis chapter. And I'm going to launch the feedback for you. Kindly give me feedback of today's class. Okay. And remember, guys, whenever you are coming for the revision class, if you read once again that chapter, if you come to the revision class means, it will help you a lot because we are solving and I am also teaching, right? If you are coming for the class for the first time you are reading this chapter means it will be difficult, right? And you can also you can ask so many questions also to me in the revision classes, right? So that's why you have to read and come at least one time. Okay. Thank you so much, guys. And can you tell me, can we go for the remaining chapters like this only, like question and answers and topic, or we can only solve the questions? Some students are there in the class. You can give me, you can tell me. Can we go like this only, first to topic and MCQ questions, or we can solve only the MCQ type of the questions? You can give me your feedbacks, which will be helpful means we will go through like that. Yes, ma'am. Topic plus questions. Less like this only, ma'am. Topic and questions. Yeah, thank you so much. So many students are telling topic plus question. I can understand you guys. You all are 12 students. You need some basic brush up, right? So that's why I'm going like this. But we are not able to solve so many questions here. But in the coming classes, we will solve. Okay. For this reason only, you have to read and come. Then we can go fastly and at least we can solve so many questions in the class. You can go through this PPT. All questions I have given is previous year questions only. Once again, you can solve those all the questions. Okay. Yeah. Thank you so much, guys.